Hi, in this video we're going to be discussing the process of transcription, which is the act of copying DNA to RNA in a process which is basically turning genes on. DNA is the repository for all information. RNA represents those pieces of information that you're using now. We'll talk about some of the molecular differences between DNA and RNA. We'll talk about several types of RNA. We'll show you the general machinery which converts DNA to RNA. That's primarily the RNA polymerase, and we'll end with talking about a few differences between bacteria and eukaryotes. On this slide we see a number of processes that are involved with the concept of gene expression, which gene expression is taking a portion of the DNA, making RNA from that, exporting the RNA from the nucleus to the cytosol, taking that RNA and making a protein, with the protein properly folded and then active. Gene expression is sometimes used synonymously, somewhat loosely, with the process of making RNA, but gene expression is more properly concerned with all of these processes. Levels of RNA depend not only on making RNA, but the rate at which it is degraded. Protein levels depend on the amount of protein that is made and the rate at which it is degraded. Once again, a misconception here is sometimes that levels of gene expression are only associated with making RNA, but you know that the level of anything is a balance between the rates that it's made and the rates at which it's degraded. Part of the reason for the bias is we know a lot more about how RNA is made and how proteins are made than how they are degraded, and that's in part because they've been studied much more, but nevertheless they are equally important. So here we see a bit of a schematic, remembering our central dogma: DNA makes RNA makes protein. We've studied previously the process of DNA replication, which is DNA making DNA. Today we'll talk about transcription, DNA going to RNA, and a little later we'll talk about translation, RNA going into protein. There's really no way around memorizing these terms. You can maybe try to remember that translation is converting a nucleic acid code of 4 bases to an amino acid code of 20, and that might help you remember that, but whatever works for you. Okay, a little bit of nomenclature when it comes from transcription, and that is there is a coding strand and a template strand. So the RNA is a duplicate of the coding strand. However, again, because of the way things are synthesized, it's going to use the template in order to produce the transcribed mRNA. If I were to give you a problem question and say, here is a piece of DNA, and just give you a single-stranded amount and tell you that it's the coding strand, you would write down the exact same sequence, but converting the T's, which is in DNA, to U's, which is used in RNA. Now we'll frequently use the expression turning genes on when we're talking about gene transcription. I think it makes intuitive sense, and it's a reminder that you have somewhere between 20 and 25,000 genes in your genome. Every cell in your body has that same set of genes, but different parts of your body uses different subsets of those. For instance, in the lung genes get turned on. There might be 10 or 15,000 of those. In the stomach genes will get turned on, and it'll be a different subset of 10 or 15,000. There'll be a lot of overlap for sure, but then there will also be genes that are uniquely expressed in one of those two organs. But remember, the stomach and the lung have the same set of genes. It's just going to express different genes to help it establish its unique character. Now here we have an example of two genes, gene A and gene B. They're transcribed at different levels, and then they're translated at different levels. A biologist would say that this gene is highly expressed and this gene is weakly expressed. There is not a linear relationship in terms of when a gene is turned on, how many copies of the RNA it will make, and how many copies of protein it will make for each copy of the RNA. But I think a typical number for eukaryotes would be about 1,000. That is, when a gene is getting turned on, it will make at least about 1,000 copies of mRNA, and then each of those mRNA will produce about 1,000 copies of a protein. So you would have a total of about 1,000,000 copies of protein per cell. I realize now that number is a little high, but that serves as an example. There is a large degree of variability of the amount of amplification from going to the RNA step to the protein step, and that is just something that one must learn on a gene by gene basis. Last point I want to make on this slide is to talk a little bit about the ratios from the highest expressed RNA to the lowest expressed RNA. In a cell as a factor, the dynamic range is about a factor of about 10 to the 4th. The dynamic range of protein expression can be about 10 to the 6th. So rare proteins can be hard to isolate in the presence of highly abundant protein. And I should say that these numbers are valid for the inside of cells. In blood, for instance, your serum protein abundances can be an enormous ratio. The most abundant proteins, albumin and immunoglobulin, for instance, can be 10 to the 10th times more abundant than the very rare protein. This slide is going to compare and contrast DNA and RNA. First, we'll look over here at the structure. You'll notice that it's single-stranded in this particular instance, not double-stranded like DNA. You have a familiar phosphate sugar backbone, a polyanion. You have sugars and you have bases attached to these sugars. The name difference, DNA versus RNA, comes from the fact of the 2' carbon. The deoxyribose in DNA, it has lost the hydroxyl group at the 2' position, so it is a deoxy, whereas ribonucleic acid has the hydroxyl group at that position. As we've mentioned several times before, the bases are slightly different. Both RNA and DNA use the bases C, A, and G. However, RNA uses uracil instead of thymine. The difference between the two structures is there is a methyl group at the 5 position. The bases are numbered starting at position 1, which attaches to the sugar 2, 3, 4, 5, and 6. Two reasons why thymine is used in DNA is one, this methyl group makes thymine a little more stable, which is good if you're trying to preserve information for long periods of time. Uracil is a little less stable, but it's a little energetically cheaper to make. It costs you something to put that methyl group in that nucleotide. Why use it if you don't need it? Now I'm going to erase these two arrows here just to show that both uracil and thymine hydrogen bond twice, and it's approximately in this direction to the base pair adenine. There are many types of RNA in cells. It's an exploding field right now, but we're going to talk primarily about 3 types. We'll talk about mRNA. M stands for message. This is the molecule that is a copy of the coding strand of DNA. There are ribosomal RNAs, which are both functional and structural inside the ribosome. They are used to make proteins while reading the mRNA code. The third RNA types are tRNAs for transfer RNAs, and these are the molecules that couple the nucleic acid code to the amino acid code. These tRNAs will bind to RNA and they'll bring the proper amino acid needed for the protein synthesis. Now the book refers to microRNAs, and there are lots of different abbreviations being used for those. These can be used to regulate gene expression, both natively used by the cell, or we can actually introduce artificial RNAs to help regulate gene expression. And we'll talk a little bit about that in the third part of the course. This is a schematic of RNA polymerase, the main protein machine that converts DNA to RNA. You can see here that the double DNA helix, which is going to be transcribed, enters through one end of the polymerase along with ribonucleotides that are going to be incorporated into the RNA. The DNA helix is unwound. We see two strands. Why don't you think for a minute which strand this would be and also which strand this would be. Okay, this is the template strand here. You're making a copy of the coding strand up here. So since both the coding strand and the mRNA are complementary to the template strand, they will be identical. The DNA is rewound as it exits the polymerase, and the RNA exits through a separate channel in the back. On this slide, we see the process of the RNA polymerase transcribing a gene. The RNA polymerase, in conjunction with the yellow protein right here called the sigma factor, which is used on most genes but not all, help bind the DNA in a region called the promoter. This is an upstream regulatory region, which we'll learn more about. Upstream of a start site, it will begin to add a few bases, and then the sigma factor is released. Now the transcription process occurs more quickly, and when it hits a special stop sequence, a sequence of nucleotides which tells the RNA polymerase to dissociate, it releases from the DNA double helix, it releases the RNA it has just transcribed, and then it can be used to transcribe another gene. Okay, in the next video we'll look at some of these we'll start by looking at some of these sequences in more detail.